Okay, so the goal here is going to be to go over metabolism generally speaking. And by that, I don't mean what I'm not going to do is go through the individual steps of every single mechanism that's happening. I'm not going to like walk you through every step of glycolysis. That's just stuff you have to kind of memorize. And there's no point in me just reading it to you. Um, so in fact, I hope you have glycolysis memorized better than I do whenever you go take your test, because that's not something I like walk around with in my head all the time. It's just not that interesting. Um, but what I am going to do is think about a bigger picture perspective on metabolism and how to integrate all these ideas together so that you don't just have to memorize every single fact. I want you to be able to regenerate a lot of this knowledge on the actual test and to think through this system from a big picture perspective, because that's what's going to allow you to answer novel questions and that you haven't seen before, to apply this knowledge to situations that you haven't seen before. First and foremost, the goal of life in general is just to propagate itself forward into the future as far as possible. And some of you have met with me and heard me say this stuff before, so I apologize if I sound like a broken record. But this is just a good way to think about the goal of life. Like, what is life? Because life is technically like a temporary defiance of entropy within a closed system. So the first thing you need for life to happen is some sort of boundary between the life and the not life, between life and environment. So you think of that as like the plasma membrane for most of the cells that are involved in this test. But then you also need a way of encoding that life and propagating it forward so that you have the same blueprint today and tomorrow and forever forward, moving forward. And so we use DNA for that. Most organisms, I think all organisms on Earth, use either DNA, some form of nucleic acid for that. So the goal of all the metabolism stuff that we are going to be talking about is just to maintain the integrity of that DNA. And by that, I mean, make sure that it doesn't get changed over the course of time. Because if you think about it, like if you take some of your DNA and then a virus comes in and inserts a new gene into it, then the integrity of your genome has been corrupted. And what is being propagated into the future from that point forward is not the same as what you are. And so that is technically evolutionary not success, right? That's the opposite of evolutionary success. So the goal of life is to propagate uncorrupted into the future your genome. In order to do that, you need to be able to replicate the genome. You also need to be able to maintain the genome. So you would think maintenance is probably priority number one, because if it gets corrupted, it doesn't matter if you can replicate it. And so you've got all of these DNA like polymerase 1 and 2 and a whole bunch of other enzymes that are involved in helping to maintain the genome to prevent errors from building up in it. You also want structures that can protect the genome against things like radiation or just other sources of entropy in the environment. And all of this stuff requires energy to be present. So life also needs a way to take energy from the environment and turn it into some sort of usable form for itself. We use ATP as our primary energy currency, but there are other means of taking energy from the environment and utilizing it to accomplish the things you're trying to accomplish. But for instance, in the kidney, there's one spot where you're able to pump sodium up its concentration gradient, not using ATP at all, but just using the flow of potassium in the opposite direction. So those are other examples of how you can harness energy from the environment and use it to defy entropy in the ways that you need to in order to maintain your genome, in order to not be corrupted moving forward into the future. However, energy is not always available for all of these things. So it's helpful to be able to store that energy in forms that can be used forward into the future as well. And the more you're able to do that, the better prepared you are to not die and to not have your genome corrupted, so that you can again continue propagating it forward into the future. I know this is all kind of like philosophical, cool, but I'm thinking about how to frame metabolism in your mind so that you truly understand what's going on here. As you're storing up this energy, though, it's really helpful if it can be doing stuff for you while it's stored as well. And that's where we think about like preference for storing food or energy as proteins rather than lipids, for instance, because proteins have a huge diversity in their structures. And so they can be used to accomplish certain things. So we've not just stored the stuff for a rainy day, but we've actually invested it into accomplishing things for us while it's being stored. It's kind of the difference between like having all of your money in a savings account versus having it in like a mutual fund where it's actually earning interest for you. It's just more efficient. But then we also want to think about if for that amount of money that we are storing long term and that we're not going to be using at all, we're just putting it in storage. We want that form of storage to be as efficient as possible. And so we and that's why we use lipids, because lipids are less heavy to carry around with us from an energy density perspective. And that's another thing that we're going to think about as we move through this. So again, that's just like an overview of the general idea here, that all of the metabolic processes are primarily maintaining the genome and also seeking to replicate as many copies of it forward into the future as possible. And then all of metabolism is how to balance our energy budget at any given time so that we're optimizing for getting the energy we need to maintain the genome and to replicate it, but also storing some of that energy so that we can do that again later in the future if we do run low on energy immediately available in the environment. So the processes, the two major classes or two major categories of metabolic processes that we use to accomplish these tasks are anabolic and catabolic processes. So anabolic processes, and many of you probably already know the difference between anabolic and catabolic processes, but I think this is the best place to really start. So I think it's super useful to categorize all of the metabolic processes as either catabolic, anabolic, or somewhere in between. Same with all of the enzymes and hormones and all of that stuff. And some of these things won't quite be anabolic or catabolic because they're oriented toward accomplishing sort of different things. Trying to think of a good example. Somebody will come up with an example at some point as we go through this. But generally speaking, you can put most things into one of those or the other category. So if we want to start thinking about anabolic versus catabolic, fundamentally, you're going to, most of you, if I were to ask, would probably say that anabolic is building stuff up and catabolic is tearing stuff down. So if you think about it from that perspective, it makes sense that you would build stuff up whenever you have plenty of resources. You know, if you're about to go bankrupt, you're not going to start building a new home, right? You only want to build things up whenever you have enough resources available so that you can do the things that you need to maintain your genome on a daily basis, but also build this new project. So anabolic processes are only really going to happen whenever you have resources available to afford them happening. So these are going to be, and I want you to all think about like different processes that would fall into each of these categories as we move through. I've got a diagram that you may have seen a little bit ago to walk through some of that, but I'm not quite there yet. So, and then catabolic processes are going to be what we use to tear down these structures that we built up during rainy days when we might not have energy super readily available from the environment, and we need to utilize some energy to continue maintaining the genome and continue carrying out all of our life processes. So if you think about anabolic and catabolic processes from that perspective, it tends to be the most helpful. However, there are a few that aren't 100% clear. So better than building up and tearing down, I actually like to think about anabolic and catabolic as either getting energy or giving energy, right? So like anabolic processes require energy input, catabolic give energy as an output. But even better than that is to think about what happens to the carbon atoms in the substrate molecules during the process. So what do I mean by that? Well, what I mean by that is that you want to pay attention to the oxidation state of a carbon atom, like your average carbon atom in a molecule throughout a process. And if you notice that carbon atom is becoming more oxidized as the process moves on, then that means that the process is probably going to be anabolic or catabolic. Does anybody have any ideas, or anybody want to say like anything in the chat? I'm just trying to make this somewhat interactive because I feel like I'm rambling right now. Catabolic processes are always going to lead to more bonds between carbon and oxygen. And the way that you can think about this is that it takes energy to reduce carbon. You get energy out of oxidizing carbon. And you can see this, we can look at any given reaction and sort of prove this point as we move through this. But if you think about the carbon atoms that you take in the form of food, so you can have fats, right, lipids, you can have carbohydrates, you can have proteins. The most energetically dense of those are going to be the most saturated fats. So the most bonds between carbon and carbon and the most bonds between carbon and hydrogen. As soon as you start trading out bonds between carbon and carbon or between carbon and hydrogen for carbon-oxygen bonds is when you start oxidizing the carbon more, decreases the energy density around that carbon, but it also gives you energy back in the form of a more usable resource like NADH or FADH2 or ultimately ATP. Anabolic processes are the exact opposite, where you are taking your carbon and adding more electron density around it. So you're reducing it. You're trading out carbon-oxygen bonds or carbon-nitrogen bonds in some cases for carbon-carbon and carbon-hydrogen bonds. And the more of that you do, the more energetically dense the molecule becomes, the closer it comes to storage, but you have to provide energy in order for that process to be carried out. So there are a couple processes in metabolism, and there's some of the most central processes that you're going to think of. If I were to ask any of you to say like what a metabolic process that you know about is, most of you would probably say glycolysis to me. And that glycolysis screws people up on this big time because everybody thinks of everybody wants to think of glycolysis as being anabolic because it's upregulated by insulin, or as being catabolic because it's breaking down glucose. And nobody's really sure like where to put it. And so whenever I make this diagram here, I always put glycolysis along with gluconeogenesis right here in the middle between catabolic and anabolic. And you'll notice in this diagram, I have all the processes in black font, all of the places where they happen in blue, and all of the rate limiting enzymes in red written out here. And then I've got some hormones in green. Normally, if I were teaching this to you one-on-one, I would ask you what some of these were and ask you to categorize all of them, but I'm just dumping this here just because I don't have a better way of doing it right now. It takes too long to make polls in these meetings. But I like to put glycolysis and gluconeogenesis in the middle here because they're the ones that will confuse you. After you take those and put them in the middle, everything else is going to fall into the category that makes sense from the oxidation reductions perspective. So for instance, in fatty acid synthesis, you're taking more oxidized carbon in the form of acetyl-CoA or malonyl-CoA or something like that, and you're building it up to be a reduced long-chain fatty acid. In the case of glycogenesis, for instance, you're taking glucose and reducing it to build up these long glycogen chains, right? And then on the other side of things, beta oxidation, obviously, you're taking these long-chain fatty acids that you just made in fatty acid synthesis, and you are tearing them down to make a whole bunch of acetyl-CoA and NADH, so high-density electron carriers, which can go on and plug stuff into the citric acid cycle, or sorry, into the electron transport chain. We'll get there in a little bit. But these are how I really like to start breaking these processes down. So back to glycolysis and gluconeogenesis. The reason that I put these in the center is, again, because glycolysis is oxidizing glucose, but it tends to get upregulated with all the anabolic stuff. And that is really just because glucose, being the primary energy currency of the body, is floating around a lot. And you need this anabolic hormone insulin to bring it into most cells where it's going to be used. And usually whenever you bring glucose into a cell, you're not going to immediately utilize it. You're going to either plug it into glycolysis or plug it into glycogenesis, one or the other, just depending on the energy available in that cell locally. But both of those processes are going to be upregulated by insulin to some extent. So just think of it in that category. And then gluconeogenesis is kind of the opposite. It tends to be upregulated in a fasting state, which is generally where catabolic processes are being upregulated. But gluconeogenesis itself, if you look at it in a vacuum, is more of an anabolic process because you're taking these more oxidized carbons and building up a glucose molecule from them. And so if you were to count up all the bonds between carbon and oxygen and carbon and carbon and carbon and hydrogen, the latter two would outnumber the former more so at the end of gluconeogenesis than at the beginning, meaning that we gain more energy density in that molecule as we build it up. Therefore, it's anabolic, even though it's upregulated by glucagon and epinephrine and all the other catabolic stuff. There's some things in here that are a little bit more confusing for people. So for instance, insulin is really easily classified as an anabolic hormone because it's released whenever you eat food. And so that means you usually have plenty of energy on board. It regulates things like fatty acid synthesis, like glycogenesis, etc. So, and it generally leads to like lean muscle building or fat building over the course of time, just depending on what your lifestyle is. But something like FSH tends to be a little bit harder for people to recognize. So if you think about what FSH does and try to think through, as I'm saying some of these things, I'm going to try to give enough of a pause for you to think through it and follow along if you do know. But I'm not going to wait forever for people to type stuff in the chat because I know that that's not the easiest thing to do. So if you think about what FSH does, it comes from the hypothalamus, or sorry, it does not. GnRH comes from the hypothalamus and causes the anterior pituitary to release FSH. FSH is a peptide hormone that then goes into the blood and stimulates the follicle in the ovary, the follicle being like the little sac that's in the ovary that holds the egg, stimulates the follicle to begin growing. As the follicle grows, so FSH is trophic for the follicle, meaning that it causes it to consume more energy and causes it to grow. As the follicle grows, it begins to release more and more estrogen, right? The bigger it gets, the easier it is to release estrogen, to make estrogen. So that causes estrogen levels in the blood to go up. And then estrogen is very much an anabolic hormone because it leads to obviously female secondary sex characteristics, but also more importantly for this test anyway is hypertrophy of the uterine lining. So we're not going to have any of this happening if we're in a starved state. Menstruation tends to turn off in women around body fat of like 12 to 15%. And so you're definitely not going to be having any of this happen whenever you're in a starved or highly catabolic state. So estrogen, it being anabolic, makes FSH pretty anabolic as well. Then and you can look at this in men too. So FSH in men stimulates Sertoli cells to undergo spermatogenesis, and that is also going to be something that obviously you don't really want to reproduce if you and all of your village mates are starving to death. So that's just another way of thinking about what FSH might be. Same with LH. In men, LH is a little bit easier to think about. It just triggers Leydig cells to release testosterone. And testosterone is absolutely an anabolic hormone. If in popular culture, not so much now, but whenever I was younger, in popular culture, testosterone was known as an anabolic steroid because all of the baseball players and all of the cyclists and all of the athletes were taking it just to be better at sports. So that's how I like to think about some of those. We could obviously go through like all of the pituitary hormones. I guess I'll talk about ACTH really quick. So ACTH being the next thing in the pituitary, like if you think about your flat peg pituitary mnemonic, FSH, LH, ACTH, TSH, prolactin, endorphins, growth hormone. ACTH stands for adrenocorticotropic hormone. And what that means is that it signals to the adrenal cortex, that's the A and the C. Tropic means that it's regulatory, and hormone, well, means it's a hormone. So ACTH goes to the adrenal cortex and primarily leads to release of cortisol from the middle layer of the adrenal cortex. So 3 layers in the adrenal cortex. Outermost releases aldosterone, middle layer releases cortisol, innermost releases DHEA, which is like a more potent version of testosterone that is more important in women than it is in men. But focusing on cortisol for a second, just like insulin is the most important anabolic hormone, cortisol is probably the most important catabolic hormone for you to know about. Cortisol, many of you will probably associate it as a stress hormone. As you get closer to your MCAT test date, your cortisol levels probably increase. The way I like to think about cortisol from a stress perspective, though, is not from like modern day stress, but more like evolutionary stress. And thinking about all of this stuff through an evolutionary lens is always going to help you, just generally speaking. But if you think about cortisol from an evolutionary perspective or stress from an evolutionary perspective, that tends to mean that you're a hunter-gatherer on the plains of somewhere and you are searching for food. So if you're in a stress environment, that means you probably haven't found a woolly mammoth or whatever in quite a while, and you and all your friends are starving. And you're probably going to be dead in the next 72 hours if you don't find some food somewhere. So if you are in that situation, what things in metabolism might be to your advantage? You want to think about things that are going to increase your chances of successfully finding woolly mammoth and getting food from it in the next 72 hours. Anything that's not geared toward that is not going to help you at all. So you want to downregulate stuff that's not going to be immediately relevant to that and upregulate everything that is. And that's how you can remember the laundry list of things that cortisol causes, because there's like 50 things in this list that you'll eventually have to know for med school. Thinking about it through this lens just makes sense of that and makes it take way less time to memorize all of them. Things like, for instance, well, since we're talking about metabolism from MCAT perspective, we want to think about the various different MCAT processes. Obviously, like everything over here, it's going to upregulate, and everything over here, it's going to downregulate. It's relative, it's really just that simple. You want to upregulate glycogenolysis, for instance, because you want to mobilize glucose in the blood, because the higher your blood glucose is, the more able you are to immediately chase something down or immediately run away from something. You also want to mobilize fats because you need more ketones in your blood, because your brain can, your brain wants glucose, but the rest of your body can operate on those ketones during that time of stress. You want to decrease things like fat buildup, right? Because that fat's not going to help you at all. You want to decrease muscle buildup because you'd rather take the ATP that you would get, or GTP technically, from translating more like muscle protein and use that to do other metabolic stuff, like just stay awake and keep being able to walk and keep being able to hunt successfully. You want to downregulate the immune system, because if you think about, well, we all learned about a lot about COVID in the last 3 years. Usually if you get an infection, you're not going to know about said infection for a few days at least, and up to a week or more at most. And so if you're going to be dead in 2 to 3 days, then it doesn't really matter if you end up getting a cold in a week. And so you downregulate the immune system as a means of shunting those resources that the immune system would otherwise require toward things that are more immediately irrelevant for finding your woolly mammoth and not dying. So that's how I want you to think about cortisol. And really, like, if you can look at everything through an evolutionary lens, that's just going to help you, generally speaking, on this test. Getting into, I'm just going to go the rest of the way through the pituitary stuff. The T stands for TSH, thyroid stimulating hormone, is probably going to be more catabolic because T3 and T4 upregulate metabolism. Then prolactin, endorphins, growth hormone are a lot less important than the rest of those. So you can look those up if you want. I kind of take a shortcut. I just want to make sure we get through all this stuff that's actually relevant for you all. So that is, from a really the biggest picture perspective, how I recommend thinking about metabolism. And then obviously, like, whenever you're studying, I recommend just drawing this stuff out a lot. Like, draw out the pathways, try to quiz yourself on this, try to redraw this Venn diagram if you want. I can post these notes with the video as well, if you want, or you can just pull up the video and look at these. But drawing out the Venn diagram, drawing out metabolism as much as you can from memory is usually, in my opinion, the most helpful way to study this. In the past, I've told people you don't need to know structures. I still don't think you need to know all of the structures, but the more people take the MCAT this year, the more I hear about it being helpful to know some bizarre structures. So if you're getting to the place where you're scoring close to, upwards of 515, above 520, that area, that's probably when it's relevant to start really learning the structures. But there you want to start probably at the citric acid cycle and then work your way out from there. You don't probably need to know the structures, every structure in glycolysis, but know the things that are closest to like rate limiting enzymes. So like PFK, rate limiting enzyme for glycolysis, right? You probably want to know the structure of phosphofructokinase, or 1,6-FPP. So just be aware of that stuff, but you don't need to memorize the exact structures. There's also some skill sets that can be useful to, if you don't know the structures, to guessing what they are. Pay attention to components of words. So for instance, like the word pyruvate, the 8 at the end tends to just mean that it is the conjugate base of a carboxylic acid, most of the time in biology. And so pyruvate has a carboxylic acid on the end. The PRYU thing tends to indicate that there are adjacent ketones, or technically like adjacent double bonds to carbon. And pyruvate has that. Its middle carbon is a ketone, and then its end carbon is a carboxylic acid. Another way to think about this is like if you think of pyrophosphate, the thing that comes off of ATP to make it AMP, and then just PPI, like the diphosphate thing. The pyrophosphate just means two phosphates right next to each other, and phosphates are just like, like kind of like carbonyls. They just don't have carbon; they have phosphorus instead. So those are just like a few tips to thinking about these things. Knowing what an acetyl group is really helpful. So like acetaldehyde, is just an acetyl group. That's an aldehyde, right? So just things like that are going to be relatively helpful as you move through all of this to predict the structures of molecules too. One more, we can say like citrate, for instance. We know now that, using what I was just talking about, we know that citrate is a carboxylic acid. So, just another example there. There's a lot more of these you can think of. If you just look at molecules and look at their names, you'll pick up more and more of the patterns over time. So I want to shift gears here and go toward this discussion that I frequently like to do with people, usually near the end of their process, that I'll just ask them, like, what happens whenever you eat food? And there's a ton of different directions we can go off of this. I haven't done it in this format before, where I'm just rambling and you all are listening. So I'm not exactly sure how it's going to go, but I'm just going to run through it really quickly. I need to take a drink too. So thinking about what happens when you eat food, frequently whenever I ask people that question, they'll be like, okay, well, you swallow the food and it goes through the acid in the stomach, and then you go through the intestine, then it gets. That's not what I mean. I want to think about like every single thing that happens. Okay, so when the first thing that happens when you eat food, and I promise we're not going to focus too hard on this because I know we want to do metabolism here, but the first thing that happens when you eat food is you chew it up. So that the reason you do that is, one, so you don't choke on it, but two, so you can taste it. And probably real two is so that you can increase the surface area for salivary amylase and lipase to get in and start the digestive process. So amylase and lipase get in and will start breaking down the carbohydrates and the fats respectively. You then swallow the food and it gets pushed down your esophagus, still being digested by amylase and lipase. Obviously, since you increase the surface area, that dramatically increases the rate at which all the stuff is being digested, because otherwise amylase and lipase could only get to the stuff that's like around the outside. So increasing surface area is huge here. Once the food gets into your stomach, there's a whole bunch of stuff that begins happening there. I'm not going to go into like every single detail, but I want to think about a few ideas that are a little bit less emphasized, but I could just, I haven't seen the AAMC like test this explicitly, some of these, but it's just like right up their alley of the kind of stuff that they like to test. So one of which is that the hydrochloric acid in your stomach serves multiple purposes. Most people will just say it breaks down the food because they're used to watching cartoons where acid melts everything. But if you've ever played with acid in the lab, you'll notice that it doesn't melt the counter, it doesn't melt paper, and it's not fun to play with at all. It's not even that cool. However, hydrochloric acid does do something that's really, really important, which is that it kills very effectively any pathogens that could be on that food that you ate. Because unless you eat your food out of like vacuum sealed bags in a lab, you probably get some bacteria and some viruses in your mouth when you eat it. Hydrochloric acid is great against that. I actually built a reputation for myself on accident, became somewhat infamous when I was in third year med school. Because one day I was in Parkland, which is, and if you live anywhere close to Texas, you know, Parkland is like the huge community hospital in Dallas. And so lots of nasty stuff happens in there. And we were at a lunch conference and I dropped my food on the floor and I didn't have time to get back in line before I had to go around again. And so I ate my food off the floor in Parkland. And there were people that heard about that like across the hospital over the course of days. I became known for that for a couple of days, which is not fantastic. But my argument back to them is that I have stomach acid and so it doesn't matter what I just dropped on the floor. Yeah, there's drug resistant bacteria in Parkland, but like we don't give HCl as a drug because that would kill you if you took a bunch of HCl as a drug. It's super effective. So I wasn't worried about it. I didn't get sick. I was fine. So primary function of stomach acid is immunity, generally speaking, because it kills stuff. Another function then this is the one that I haven't seen really tested but I could see it tested, is that just like we chewed up our food in the mouth to increase access for salivary amylase and lipase, it's also helpful to do something like that in the stomach too. But whenever we do it in the stomach, we're doing it at the level of the molecules rather than the level of the entire bolus of food. So whenever hydrochloric acid accesses the food that's made it down to your stomach, it's going to take the proteins that are in that food that are in their globular form still probably, and it's going to completely denature them. The benefit of that is that you're also releasing pepsin or pepsinogen that gets activated to pepsin in the stomach that will start cleaving peptide bonds. However, pepsin is a huge enzyme itself, right? And so it's not going to be able to get into those interior hydrophobic amino acids unless you denature the thing, stretch it out into its long chain form, complements of HCl, and then pepsin's able to come in and start cleaving, hydrolyzing those peptide bonds so that you can break down the food like exponentially more effectively than you could if you didn't have hydrochloric acid present. So that's just something to think about from the hydrochloric acid perspective. You do also need to know that parietal cells are what's responsible for making HCl and that chief cells make pepsin. There's some other stuff that happens in the stomach as well, but here we're primarily focusing on metabolism, so I'm going to move on. After you get out of the stomach, you go through the pyloric sphincter to go from the stomach into the duodenum or the duodenum. Technically, duodenum is the correct pronunciation, but I think it sounds weird. So I always learned duodenum as I was learning this stuff. So we'll stick with that for right now. The duodenum is the first of three segments of the small intestine. The next one's being the jejunum and the ileum. But the duodenum is probably the most important component of the GI tract for our purposes here, because you start to get absorption happening, but you also have most of the digestion that's going to happen in the entire process happening here. And the reason for that is because there's this little window in the duodenum where you, with this thing called the sphincter of Oddi, where the pancreatic juices as well as stuff from the liver and the gallbladder all get dumped in together. And the pancreas is responsible for most of your digestive enzyme production. So the pancreas releases a whole bunch of and I think I have a picture of this prepared as well. I'm going to go ahead and pull it in. But the pancreas releases a whole bunch, and this is what I'm talking about, this duct here. Pancreas releases a whole bunch of peptidases that primarily trypsin, but a few other chymotrypsin, a bunch of others, as well as lipases, and all of that stuff just gets in and starts breaking up the food even more significantly so that we can get down to really small molecules and be ready to start absorbing those. I'm going to talk about here in a second how each of the molecules gets around in the blood, because up to this point, we've had everything kind of meshed up together, but we're going to start separating out carbs from fats from amino acids here in a second. We also have juices from the gallbladder and from the liver coming down as well, which are incredibly important for digesting lipids. We are going to talk about that maybe at the very, very end today, but suffice it to say that the juices from the gallbladder are made up primarily of cholesterol that has had carboxylic acid groups added to it, which makes it not just hydrophobic, but also having some hydrophilic components. And that's super important for this process of emulsification. Emulsification means that it's basically taking these fat droplets, because fat is going to all clump together in the GI tract with the solvent being water. Fat will all kind of clump together and stick on itself and ends up getting a very large volume to surface area ratio, or very small surface area to volume ratio. Just like we talked about in the mouth with chewing the food up, and in the stomach with denaturing the proteins, we need to increase the surface area to volume ratio here for these lipid droplets for them to ever be absorbed efficiently at all into the GI tract. And so the emulsifying force of the cholesterol, the bile acids comes in. We've got the cholesterol parts that will cozy up with the lipids, and we've got the carboxylic acid groups that will be hydrophilic. And so you can get in and start breaking these lipid droplets up into much smaller, meaning more surface area per volume components, so that they can then be cut up by lipase, which is a hydrophilic enzyme, right? All enzymes are going to be hydrophilic as long as the solvent is water, because they're going to arrange themselves so that you have hydrophilic residues on the surface and hydrophobic residues on the interior. And so in order for pancreatic lipase to get in here, we need to pull these fat droplets into the smallest things that we possibly can, so that pancreatic lipase can start digesting it. So this process of emulsification by bile is just hugely important. And like I said, we can come back and talk more about cholesterol and the things it does in the body and bile at the very end if we get to that. I don't know that we end up getting to that here, though, because I have a whole bunch of stuff to talk about. Anyway, so once that happens, the lipids is relatively easy for them to get across the GI lining. They don't need super specific transporters or anything like that, because they're hydrophobic. So they can just migrate on through the wall, and they get into the lymphatics and the venous blood and will make their way up from there. Trying to think. Let's go ahead and follow those. So those things get in and get into the blood and will hitch a ride on a protein. The protein, once the protein has the lipids attached to it, it's going to be called a chylomicron. The reason that the protein needs, or the reason that the lipids need a protein to carry them around, because this isn't the case for the carbs or for the amino acids at all. The lipids need a protein to carry them around, though, because they are not hydrophilic, so they're not going to go into solution in blood. If you've seen the OCHEM webinar I did, I mentioned this rule of figuring out how many hydrogen bond opportunities a molecule has versus how many carbons are on it. And if you count that up for cholesterol or for any of the other lipids, it's not going to be close. They're definitely hydrophobic. And so if you have a bunch of hydrophobic stuff floating around in your blood, that's fine when you're in the aorta. But as soon as you get into small vessels, they're going to clog up and you're going to have a heart attack or something like that. So generally speaking, we want to throw these things onto proteins because proteins are hydrophilic enough to pull these lipids into solution with them. The chylomicron is the most fatty of these lipoproteins. They're called lipoproteins because they're proteins and lipids, right? The chylomicron makes its way up to the liver and drops off all these proteins for repackaging there. And you'll usually repackage them in that spot as LDL, but you can make IDL or HDL. In all of these acronyms, the D and the L stand for density lipoprotein. And so HDL is going to stand for high density lipoprotein, IDL for intermediate density lipoprotein. You also have a LDL for low density and a VLDL for very low density. And there's actually like a host of other ones as well, but those are just the ones that you have to know for this test. Just to make more sense of what those names stand for, because this used to annoy me because I felt like it wasn't explained very well at any point. The density of a lipoprotein just refers to the actual density of that molecule. So, like the density of water is roughly 1 gram per centimeter cubed, right? The density of a lipoprotein. So if you think about the components of it, you've got protein and you've got lipid. Usually proteins are going to be close to the same density as water, whereas lipids are going to be a lot less dense. So lipids are usually going to float on water. So as you increase the lipid that is bound to your protein, the density of the lipoprotein is going to go down, right? So the lowest of the density lipoproteins is the one that carries the most lipids per protein. The highest density is carrying the least lipids per protein. And so, if you've ever heard about good cholesterol or bad cholesterol, and I think some of that's being debunked actually, but if you think about it from a fundamental perspective, good cholesterol is going to be the cholesterol that's the least likely to give you an ischemic event. So, the least likely to cause a heart attack or to cause a stroke. And the way in which cholesterol causes heart attacks or causes strokes is by precipitating out of the blood. So if you have the highest density lipoproteins, meaning the most protein per lipid, you can think that the probability of that thing accidentally coming out of solution or the solution becoming super saturated with that is going to be really, really low compared to LDL, which has the most lipid per protein that you could possibly have. And so it's just going to have a way lower KSP, right? And so this is how we can start thinking about applying general chemistry knowledge, even though we're over here talking about biochem. So all of those density lipoproteins will eventually leave the liver. And the reason they had to pass through the liver is because on the way up from the GI tract, everything has to go through the liver on the way up to get back to the heart. So they'll eventually leave the liver and go to the heart. And once they get in the heart, then we're going everywhere in the whole body. And so then depending on the metabolic state, meaning anabolic or catabolic, you'll either pull those lipids off of the lipoprotein and into the cells, or you'll take lipids from the cells and dump them onto the lipoproteins, increasing their density and sending them back to the liver for storage. This is super complex, determining which one's going to happen. So usually on the test, I would predict that the passage will probably tell you. if then, and even if they don't tell you explicitly, they'll say something like the levels of certain hormones or whether you just ate food or something like that. So that's just how to think about that stuff. Once the lipids is kind of nice whenever they actually get to the place where they're going. So here we've spent all this time talking about how they get transported around, but when they get where they're going, it's usually pretty easy to move them into the cell because again, they're largely hydrophobic. So if we back up and think about, so that's how the lipids get to the cell. We'll talk about what happens there here in a second, but we got to get everybody else to the cell first. So if we back up and think about the amino acids and the carbohydrates, as they're getting chopped up in the GI tract, they're going to start getting absorbed once they're in either their mono peptide, dipeptide, or monosaccharide or disaccharide states. So that's where these get absorbed. Lots of them have really specific transporters. I've forgotten way more about this than I know right now. It's not super relevant for the MCAT, but they have specific transporters that will pull them across from the GI tract into the blood or into the lymph. And at that point, they can just travel easily because they are not going to come out of solution. So they're water soluble and they'll just float around in the blood. They have to go through the liver on the way back up, but eventually they make it to the heart and they get pumped out to all the rest of the body. In the case of amino acids, those are usually going to be relatively easy to get into the cells. You have all these specific transporters that will pull specific things in based on whatever the body needs in that certain spot at a given time and how much availability there is in the blood, as well as how much there is in the cell. But in the case of the carbohydrates, it's a little bit more complicated to get them into the cell. And that's what I want to spend just a little bit of time talking about here. So I'm sure that you are all at least peripherally familiar with the fact that there are multiple different types of glucose transporters. You've got GLUT2 and GLUT4. There's also a GLUT1 and a GLUT3, and I think there's a whole bunch of other ones too. But the ones you need to know about for this test are GLUT2 and GLUT4. And just know GLUT1 is brain. Maybe have that in the back of your knowledge base as well. So the first thing that happens in order to get glucose up into your cells is that on the way through the liver, it's going to just go into the liver cells. The reason for that is because the liver has what we call GLUT2 transporters that are always going to be present in the membrane of the cells and are always going to pull glucose down its concentration gradient. So if you have more glucose in the blood than you have in the hepatocyte at the time that blood makes it to the liver, then there will be glucose influx. So glucose is just going to go straight through into the hepatocyte through the GLUT2 transporter. And at that point, it's going to be phosphorylated by this enzyme called glucokinase. Glucokinase has a really high Km, meaning that it has a really low affinity for glucose. So it's only going to phosphorylate glucose at high glucose concentrations. Once the glucose is phosphorylated, this is important, we'll come back to it later, but once the glucose is phosphorylated, it will either get stored as glycogen or used up in glycolysis. The liver needs energy too, and so it will do some glycolysis, but most of the glucose that gets into the liver is going to be stored as glycogen and then later released whenever glucose levels in the blood drop, such that now for GLUT2 to send glucose down its concentration gradient, it's taking glucose from the cytosol of the hepatocyte and putting it back into the blood. that's whenever you get to a relatively low blood glucose state. So the way that I've told people to think about the liver and how it handles glucose before is it's like the mom of all the other cells. So it's just like taking care of everybody else. It's going to eat too, but I'm not going to be like crazy about it. And it's just going to make sure everybody else has glucose, specifically the brain. So that's the first place that you interact with glucose using a glucose transporter. I said that the Km of glucokinase is high. So is the Km of GLUT2. These are both relatively high compared to their counterparts, which we'll talk about in a second. What that means is that whenever glucose is low in the blood, GLUT2 doesn't want to interact with it. It's not going to pull glucose into the cell whenever glucose is low in the blood. We need high glucose in the blood in order for GLUT2 to start taking some out. And that's another way in which it's like the mom of the rest of the cells from a glucose perspective. And similarly, whenever you get glucose into that hepatocyte, glucokinase isn't going to start phosphorylating it yet until the glucose concentration gets to a certain level, because it needs to make sure that glucose doesn't need to immediately leave and go back into the blood, because phosphorylation is kind of like the cell claiming the glucose as its own, right? As soon as you phosphorylate the glucose, it can't go back out of the transporter anymore until you dephosphorylate it. And so you need to make sure that you actually want to keep it before you phosphorylate it. Then once the glucose makes its way to the heart and is pumped out to the rest of the body, we encounter the second instance of GLUT2 before we can do anything else. Because right now, at this point, now that we've pumped the glucose out to the rest of the body, you do have glucose coming up against, via the bloodstream, but coming up against cells that need it. However, those cells haven't yet gotten permission to take up the glucose. The reason for this is because we need sort of a centralized mechanism for controlling when cells are and are not allowed to take up glucose. Because if they were all allowed to take up glucose however frequently they want, you would just die immediately. The reason for that is there's at any given time, there's only ever about 1 or 2 teaspoons of glucose in your entire bloodstream. Like all of the blood in your body is like 8 liters. You have like 2 teaspoons of glucose in that whole volume of blood. So you have to be relatively judicious with how you take up that glucose. So the way that and you all probably know where I'm going with this, but we want to back up and think about glucose interaction with the pancreas before we can think about it getting into any of these other cells that are going to utilize glucose. So the pancreas is the second instance of GLUT2 and glucokinase. Once the glucose reaches the pancreas, it will go through these GLUT2 transporters just like it did with the liver and begin being phosphorylated by glucokinase. The presence of phosphorylated glucose in pancreatic beta cells indicates to those cells that there is a high blood glucose concentration, because phosphorylated glucose isn't getting there unless there is a high blood glucose concentration because of the Km of GLUT2, right? High Km means we're not going to pull glucose in until we get relatively high glucose in the blood. And then high Km on glucokinase means we're not going to phosphorylate it until the glucose we've pulled into the cell is also really high. So there's like 2 steps to make sure that glucose is actually high before the pancreatic beta cells start doing their thing, which is releasing insulin. One thing that the MCAT, and this is kind of a curveball, but one thing that the MCAT will like to ask about is where things like insulin get made. And yeah, beta cells, sure, great, fantastic. But what they're really going to be testing you on is everything prior to that happens within the beta cell. So you've got a gene for insulin, and that gene is going to get transcribed in the nucleus, make mRNA. Then your insulin mRNAs, or your insulin hnRNA, heteronuclear RNA, is going to get processed into insulin mRNA. That's going to make its way to the endoplasmic reticulum, not the free ribosomes, but endoplasmic reticulum, because we know it's destined for secretion, which means it has to be translated on the ER. From the ER, it's going to be shipped over to the Golgi for processing, meaning sometimes glycosylation. I don't think insulin gets heavily glycosylated, but you can glycosylate, meaning add sugar groups to proteins to just modify them to add more diversity before you secrete them. You can also cut off specific things like, for instance, translation always starts with a methionine residue because that's what the AUG codon codes for, and that's the start codon. However, most amino acids don't have methionine as their N-terminus. So that means that you cut it off in the Golgi in processing. You can, with glucose, you also cut off this like C-peptide component of it. There's a couple like segments of glucose that get cut up, and then the whole big thing gets spliced together at the end. And I think I said glucose, I meant insulin, so I apologize for that. But the whole thing gets spliced together at the end of processing and then shipped in an endosome up to the surface of the cell, where that endosome then merges with the surface of the cell, and now insulin's been released. So we just released insulin from the pancreatic beta cell as a result of a high concentration of phosphorylated glucose in the cytoplasm of that cell. And so now you've got this insulin floating around the blood. Fortunately, that's a protein. And so it doesn't have to be transported by another protein. It can do its own thing. And it starts to make its way out to the cells. Once that insulin binds to the insulin receptor, and this is for like all of your other cells that we haven't talked about so far. So we've ruled out brain, we've ruled out liver, we've ruled out pancreas. Pretty much everyone else has these transporters called GLUT4 transporters. And these are, we talked about the liver being like the mom of all the other cells. This is like completely the opposite of that. This is like the younger sibling of all of the other cells. It wants everything. It does not want to share at all. So the GLUT4 transporters, sorry for those of you who are younger siblings, I'm not meaning to be too mean, but the glucose, so the GLUT4 transporters are not going to consider anyone else, meaning they have really low Km values, meaning super duper high affinity to glucose. So anytime that they are interacting with the glucose, they're going to be grabbing it up and pulling it in. All right. So you keep these GLUT4 transporters sequestered inside vesicles all of the time unless insulin is bound to the receptor, because insulin is only going to be bound, hopefully, whenever we have high blood glucose concentration. All right. So GLUT4 lives inside of these vesicles that are sequestered in the cytoplasm. You can see the drawing here. These vesicles were made in the Golgi, right? Because GLUT4 was made in the same way that I just talked about insulin being made. And so it just gets embedded in that vesicle, and the vesicle blebs off of the Golgi and just hangs out in the cell until we get this signal from insulin. And it's really complicated, like Jack kinase thing. We don't need to go through that. But as soon as insulin binds its receptor, it stimulates the merging of these vesicles with the plasma membrane of the cell, such that now the GLUT4 transporters are embedded in the plasma membrane, and they can start pulling glucose down their concentration gradient. These cells also have a different version of glucokinase. So I talked about glucokinase being the thing that phosphorylates whenever you're in the liver and in the pancreas. But all of these cells that have GLUT4 have another enzyme called hexokinase, which is also greedy, just like GLUT4. So these both have low Kms, meaning super high affinity, meaning they're going to pull in and then phosphorylate glucose no matter what the glucose concentration is. So one of the most effective ways to accidentally kill somebody in the hospital is to give them insulin whenever they don't need insulin, because that's going to cause, the insulin's going to cause all these GLUT4 transporters to go up and merge with the plasma membrane. And so say you haven't eaten in a few hours, your blood glucose is relatively low. If you start pulling all that blood glucose into your cells, the total body water in your, or the total water in your cells is like 2.5 times the total water in your vasculature. And so if you start pulling all that glucose from your bloodstream into your cells, you're going to tank your blood glucose immediately. And that's super easy way to die. So that's why this mechanism is highly regulated. All right. So that's really how glucose gets into the cells, and then it gets phosphorylated once it gets in. I guess we could just go ahead and talk about what happens to the glucose since we're already here. So when glucose gets phosphorylated, I mentioned a second ago, it can go one of two ways. It can become glycogen, which means just stored for much later. And that's only going to happen if insulin is there, and if glucose concentration and acetyl-CoA and ATP concentrations in the cell are all also high at that time. Otherwise, we're not going to start making glycogen if ATP is low, or if acetyl-CoA is low, or if glucose in the cell is low, because we need to go ahead and use that glucose to maintain the stuff that we're doing, maintain the integrity of the genome, maintain the integrity of our membrane, all of that stuff that I mentioned right at the top of this meeting. So let's say we do glycolysis first. So that means that there's already a lot of glucose in the blood. So let's say we do glycogenesis first. So the main two things I want you to know about glycogenesis, and we can talk about some of this later, but primary two things are glycogen synthase, right, where we're forming the 1,4 glycosidic linkages here, and branching enzyme, which is where we form the 1,6 glycosidic linkage. I think you can see how this plays out. Obviously, these things become giant sheets of glycogen, not just like the strand I've drawn here, but I mean, this was enough. So if you want me to draw more, we can do that later. So that's the whole thing with glycogen. Once you get into a catabolic state, you have glycogen phosphorylase and debranching enzyme that just do the exact opposite and tear down glycogen. Some people might wonder why we make glycogen. Why don't we just let all of this glucose phosphorylated float around in the cell until we need it for glycolysis? And the reason for that is because by putting into these large sheets, you minimize the distance between the glucose molecules, which therefore minimizes the amount of water that gets between the glucose molecules. Because if you're thinking about, we want to store energy for later, that's really important. But we also talked about how it's even more important that the energy we're storing for later be somewhat functional or at least not detrimental. But if you're carrying around a lot of glucose molecules as individual glucose molecules, each one of those is going to get a ton of water molecules forming a solvation barrier around it. That's just how solubility works. And you're going to end your glycogen would be, or sorry, your glucose that's not glycogen would be incredibly heavy. You could not walk around. If you're a tree, that's fine, but you're not. So don't do that. So we store them as these giant sheets so that we just get a solvation barrier across the surface and on the other surface and on the edges, but not throughout the whole thing. It just makes it far lighter to carry. So we only keep it in its individual subunits whenever we're getting ready to transfer it from glycogen to glycolysis or vice versa. Okay, so that's glycogenesis. Once we get to the process of glycolysis, we've already been phosphorylated, so we go through the next step of phosphorylating the glucose 6-phosphate, and that can send us down the glycolysis pathway, where we eventually split the molecule and we end up with 2 pyruvates at the very end. And again, like I said, I'm not going to go through every step of those things. There's also a third thing that you can do from the perspective of glucose 6-phosphate, and that many people will tend to forget, that is actually probably the most centrally important thing that we have to think about whenever it comes to metabolism, because remember, the purpose of metabolism was to maintain integrity of the genome first and foremost. And so that is production of, so taking glucose 6-phosphate and then running it through a dehydrogenase to get some NADPH out, right? So by that, what I mean is the pentose phosphate pathway. So you're taking glucose 6-phosphate and ultimately making ribose and NADPH. Ribose is really, really important for integrity of the genome and propagation of the genome because it is the backbone of DNA and the backbone of RNA, right? So we definitely need to do that. It's also important to be able to make NADPH because that is your primary reducing molecule in the cell that is really important for cleaning up oxygen-free radicals, but also for synthesizing fatty acids in the fatty acid synthesis pathway. So don't forget about that. I'd say that one's not quite as high yield as some of these others, but just be aware that it makes ribose for nucleic acids. It also makes NADPH as a reducing agent. We could talk about G6PD deficiency, but I don't think that that's relevant right now. G6PD deficiency is just a disease where you don't have 2 good copies of that enzyme, so you can't make as much NADPH, and so your red blood cells end up not having a very good lifespan because of that, because red blood cells carry a ton of oxygen around, and so it's just really, really, really important for them to be able to clean up oxygen-free radicals as a result of that. So anyway, so that's just what's going on there. Once we get through glycolysis and we've made the pyruvate, we take that pyruvate and turn it into acetyl-CoA, and then eventually that gets plugged into the citric acid cycle. One thing that I tell people frequently is that the citric acid, if I had to pick one pathway for you to know, it would probably be the citric acid cycle. You all have this picture in your biochem resources folder. I think it's number 3 in your student folder that you have with us. This is also on Google, but this is just my favorite citric acid cycle picture because it color codes everything. So it shows you what the catalysts are, what the outputs are, and what the inputs are. Paying particular attention to the outputs is super important for this. So you see it starts over here, which is the only thing I don't like about it. I wish it started at the top. But it starts over here. You take that acetyl-CoA and combine it with oxaloacetate to form citrate. And then you see a few more steps. And then eventually we start producing NADH and giving off CO2. Because we're giving off CO2, this has to be a catabolic process, right? Because CO2 is the most oxidized form of carbon. And so if we're taking anything and making CO2 with it, definitely catabolic, means we're definitely getting energy out of it. The energy we get out is primarily in the form of NADH. We also get some FADH2 and some GTP, which can be transferred out for ATP. But most of what I think is super important for this mechanism is just being aware of these 2 steps right here, the isocitrate dehydrogenase and the alpha ketoglutarate dehydrogenase steps. Those produce CO2. And so you've got carbon biomass leaving, which is just important to be able to track. And you're also making 2 energetically dense electron carriers that will eventually plug into the electron transport chain, which we can talk about later. So that's what glucose metabolism looks like. We also will get, we end up sending the intermediates from beta oxidation into this same pathway, right, as acetyl-CoA. And so we end up using this pathway for that as well. You can also take things out of this pathway and synthesize amino acids or synthesize proteins from those amino acids. You can also take things out and synthesize glucose via gluconeogenesis, right? If you take oxaloacetate, for instance, you can go plug that back into gluconeogenesis. So the reason I think this is the most important pathway is because it's really the central hub for all of metabolism. It's where you take things and make other things out of it, right? So like I tell people, a thing I did at one point when I was in med school is that I went on a Cliff Bar only diet for like 3 months. I think sorry, I think it was just 1 month, but whatever. I went on a Cliff Bar only diet for an absurd amount of time where I was just eating chocolate chip Cliff Bars. It's not great for you, but I was also taking a multivitamin. And so what that did was it caused my metabolism to be super inefficient because I had to just convert all the garbage that was in those Cliff Bars into the things I actually needed. And that's just like a fantastic way to lose weight. So don't do it, but that's what I did and it worked. I just wanted to prove a point that I could not die if I did that for a long period of time. And that's because the citric acid cycle is just super effective at interconverting these things. So we talked all the way through for glucose. We made it to the cell with the lipids, and then obviously beta oxidation is how we get things into the citric acid cycle from here. We haven't talked about the amino acids at all, and this tends to be something that people gloss over a lot that doesn't get taught in your classes most of the time. And it's not the most important thing ever for the MCAT, but I just want to make a couple of points on it. So once the amino acids, now they floated out to the cells and they get into the cells a little bit more easily than glucose does. You don't have to worry about insulin and GLUT4 and all of that stuff. They get into the cells and then they're doing their things in the cells, and they're going to be constantly in and out of the citric acid cycle as your body needs more or less of certain types of amino acids or more or less energy. So you can, if you're really, really starving, you tend to lose a ton of muscle mass because you can break down those huge amounts of protein in the muscle and turn them into amino acids and plug them into the citric acid cycle to get either glucose or ketone bodies out of them. How to determine whether an amino acid is glucogenic or ketogenic can end up being a really important thing on this test. You may have memorized that all of the amino acids can ultimately become glucogenic, except for leucine and lysine. But I just want to show you this picture, and this is also in your biochem resources folder to sort of explain why. So you see here in this picture that all of the amino acids in yellow are ketogenic and all the ones in pink are glucogenic. One thing you might notice if you're being really astute when you look at this picture is that the ketogenic amino acids are all coming into the citric acid cycle as acetyl-CoA, whereas everything else can be either way, right? It can be glucogenic or it can be ketogenic, but there's no real like rule after acetyl-CoA. The reason for this is because the first 2 steps of the citric acid cycle, which we have up here, or I guess the second and third step, I don't count the conatase, but these 2 steps, you lose carbon in the form of CO2 in each of these 2 steps. And so acetyl-CoA or molecules that come in prior to these 2 steps as acetyl-CoA are going to contribute 0 biomass ever to glucose, right? Because if you come in as acetyl-CoA, you lose 1 carbon in isocitrate dehydrogenase and then another one in alpha-ketoglutarate dehydrogenase. So if you come in up there, both carbons are already gone before you ever get around to oxaloacetate, which is the way that you would ultimately end up forming glucose from the glucogenic amino acids. So everything that plugs in here is alpha-ketoglutarate or after can go form glucose because it comes in after, therefore it can contribute carbon biomass to glucose. These other things can't. So it's just leucine and lysine that fall into that category, just so you know. I would pay attention to these probably right here are the most likely to be tested, specifically glutamate and glutamine and alpha-ketoglutarate. Those are all like pretty structurally similar. And so just know that that's where those plug in maybe, but it's a super low yield idea. I just wanted you to be aware of like how to predict whether an amino acid was going to be glucogenic or not glucogenic. And then they can technically all be ketogenic because if they come in as acetyl-CoA, they're basically there already because acetyl-CoA is a ketone. So that's just really one idea on amino acid metabolism. The other major idea is just if you're thinking about what the anabolic equivalent of that is, I mean, well, it's just like protein translation, which you learn about ad nauseum. I think that I've got a video that goes over it pretty thoroughly in your whiteboard videos folder, so you can check that out. Let me know if the video file is corrupted too. I know that that's happened to a few people lately. I made a spreadsheet pretty recently where I've got all of the links to all of them. Compliments of Samir. He's the one who let me know. But anyway, so let me see where we were going to go from this. So we did the amino acid metabolism. Next thing I have done here is to talk about the electron transport chain. The AAMC doesn't test this as heavily as it used to, but it still tests it probably too heavily, in my opinion. So I just want to go through it briefly. I'm not going to go through every single step, all of the intermediates and carriers and stuff. I do recommend knowing those, unfortunately, but Again, that's just something that you want to write out. I don't have a ton of value to add on like thinking about how complex 1 donates to 3 and how 2 donates to 3 and where cytochrome C and all that stuff are. That's just stuff you have to memorize. It's not going to help on your thinking end of things. What I really want to talk about is how this thing actually works. So if you think about the electron transport chain, you're taking high density electron carriers like NADH and FADH2 and plugging the electrons in, and then you have a sequence of redox reactions that send those high energy electrons down to oxygen ultimately. And at the same time, those favorable reactions, and they all have to be favorable, are coupled to the pumping of protons across the mitochondrial inner membrane to build up a gradient, a pH gradient, really, from the inner membrane space down to the mitochondrial matrix. This gradient can be dissipated through ATP synthase, which is what powers the production of ATP. I know you all know that already. I just wanted to summarize it briefly. But what I think is most interesting and where the MCAT's most likely to test this is in ways that you can mess with it. Because normal functioning is not hard. It's not hard for you to memorize that stuff. It's not hard for you to answer questions on it because you can just memorize it. But once we start throwing kinks into it, is where things can get somewhat complex. There's really 2 toxins that I want to talk about here specifically, just to shed more light on how this whole process works. The first of which is cyanide. So cyanide is a complex 4 inhibitor. By complex 4 inhibitor, I mean that it binds to complex 4 and prevents complex 4 from being able to take electrons from complex 3. So by inhibiting complex 4, you cause all these downstream effects that ultimately lead to killing you if you have enough cyanide, right? Because if you just inhibit one complex 4, you're not even going to know about it because you have, maybe trillions of cells and each one of them has a lot of mitochondria and each one of those has a lot of complex 4s. But as you get more and more cyanide, then you're going to get a greater percentage of your complex 4s inhibited and therefore less and less ability to transfer those electrons off of complex 4. Even though you can't use complex 4, you're still pumping out quite a bit of protons though. And you can still generate enough of a gradient to create ATP from that, which is fantastic. However, complex 4 being inhibited means that complex 3 is going to start becoming pretty reduced, right? So you're going to end up with a lot of reduced complex 3. And with complex 3 being reduced, the equilibrium and the reaction between 2 and 3 and between 1 and 3 has now been shifted back to the left so that you start to get a whole bunch of reduced complex 2 and a whole bunch of reduced complex 1. Once you get a bunch of reduced complex 1, you can't donate electrons from NADH to complex 1 anymore. Therefore, you get a huge buildup of NADH. All of these mechanisms that we look at here require NADH to be a product. And so as NADH builds up, you shift the equilibrium of all of those reactions back to the left side. So you get a huge buildup of NADH, and it makes these reactions not able to happen anymore, basically, because you can't take your NADH and turn it back to NAD plus through these reactions, because they're favorable in the direction we're showing them. They're unfavorable in the reverse direction. So they're just not going to happen that way. And so what that does is it inhibits. So now that we've inhibited complex 4, that caused complex 3 to not work. Complexes 1 and 2 don't work anymore. And so we just have NADH build up. We run out of NAD plus, and therefore we can't run the citric acid cycle anymore. We can't run beta oxidation anymore. We can't run glycolysis anymore. And that's why we die from cyanide poisoning. So not a good thing, generally speaking. So I just want you to, whenever you're thinking about all of these pathways and all of these mechanisms, it's really useful not just to try to memorize every step, but to think about what happens if I mess up one of these steps and just go through it slowly with yourself. And if you're not sure whether you got the right answer or not, please check it with us. Like every single one of you has at least one tutor. So you can just send it to us and we'll say, yeah, that's right. Or no, that's not right. Think about it this way. You can send it to me too, but I get a lot of messages right now because MCAT season's kind of at its peak. So I'm kind of slow to respond. All right. So thinking about back to the electron transport chain, I now want to think about what happens when we do something just a little bit different from inhibiting complex 4. Specifically, what I'm talking about is an uncoupling agent or a decoupling agent. So there's a lot of different ways to do this, but the decoupling agents, fundamentally, their job is to dissipate the gradient of protons without allowing them to go through ATP synthase. So one of the most common ways of doing this is just putting channels in the membrane that facilitate diffusion of those protons back down their gradient into the matrix. I can't remember what bacterium it is. You'll learn about it in med school. There's a bacterium that does that specifically. We've also got a bunch of antifungals that do this to fungus. not so much to us anymore. And there are a whole bunch of antibiotics that do this to bacteria. So this is a good strategy. Everybody's using it against everybody else in the circle of life. the Another way to do it is just to take a small molecule that can pick up the protons from the intermembrane space and drop them off in the matrix. I can show you what that looks like as well. But with the idea here is that you're decoupling the electron transport chain from oxidative phosphorylation. So these molecules were used as really, really popular weight loss drugs in like the 70s and 80s, and I think maybe even early 90s, actually. And you can still find them, but don't. You'll probably not end up in a good spot. The reason that they're used as weight loss drugs is because by decoupling the electron transport chain from oxidative phosphorylation, from ATP production, what that does is it's not going to cause you to run out of ATP. This is not how people die who overdose on these. What it does instead is it upregulates the electron transport chain, because if you have too little ATP, that's like one of the most potent negative feedback inhibitors in the entire cell. So if you don't have enough of it, you're not going to get as much feedback inhibition. So you're just going to upregulate all of your other catabolic processes, right? So you upregulate electron transport chain, upregulate the citric acid cycle, beta oxidation, glycolysis, not fatty acid synthesis though, because that's the wrong direction. But so upregulate all the catabolic stuff whenever you have less ATP. And so by doing that, what you're doing is effectively burning more calories to make the same amount of ATP. This works amazingly, which is why people marketed them as weight loss drugs. However, the problem is that the therapeutic window is relatively narrow. Therapeutic window refers to the minimum amount that you need to get an effect versus the minimum amount to kill people. And so the therapeutic window for these drugs are just really narrow, but it has nothing to do with you running out of ATP, because again, like I said, you have lots of mitochondria. And so even though you're inhibiting you're dissipating some of the gradients in other cells won't get completely dissipated, and even the ones that are going to be just dissipated a little bit. It's not like all the protons are going to be gone. So if you don't die from running out of ATP, think about how you might. What might the effect of this be? The effect is that by instantly upregulating all of your metabolism, you're causing stress on some of your other physiologic systems that would otherwise cope with this. So if you think about things you might do to burn calories if you don't have a decoupling agent, but you still want to eat food, you might think about going and running around, or going and lifting weights, or doing a bike ride, or swimming, or something active, right? Whenever you do those active things, the reason that somebody who's 400 pounds doesn't start doing that immediately and do it at like all day long on their very first day is because they're not conditioned to do it. So they get their legs burned, their arms burned, their lungs burned. They don't feel fantastic. Nobody does when they start working out. I can speak to this firsthand, and so that makes you not want to do it. The reason that you feel that way is because you've got acid building up in those tissues as you're doing this, and it activates dynorphin receptors, which mean pain, right? Sometimes you get endorphins, which are going to counteract that, but mostly you're still going to feel like it doesn't feel great. Now, so as the acid builds up, you increase your respiratory rate to get rid of that acid. You increase your heart rate to pump more of that acid back to the lungs faster so you can respire more of it out. If you're somebody who doesn't work out at all, your respiratory capacity and your cardiac pumping capacity are probably lower than like Lance Armstrong in the 90s or whatever. And so if you suddenly start burning the same number of calories per day that he does on the Tour de France, even though you're sitting around on your couch, which is what these people do, then you're probably not going to be able to handle the acidity in your blood as a response, right? Because every time you run something through the citric acid cycle, you produce 2 molecules of CO2 every single time. Every time you go through glycolysis and make acetyl-CoA, you produce a molecule of CO2. So for every glucose you put all the way through metabolism, you're making 6 CO2s doing efficient metabolism. 6 CO2s every single time you do this, and now you're having to do it at a rate that was 5x your normal metabolic rate. You're going to be making a lot of CO2. That CO2 then diffuses straight out of the mitochondrion, in the case of the citric acid cycle, straight out of the cytoplasm into your blood. It's going to diffuse into your red blood cells and react with water in a reaction catalyzed by carbonic anhydrase to make carbonic acid. And then that carbonic acid is going to dissociate to some degree and form hydronium plus bicarbonate. And then those are going to go over to your lungs and then turn back into CO2 as the equilibrium shifts back to the left, and you're going to breathe out that CO2. But in the case of somebody who hasn't been doing the Tour de France or training for it, if they suddenly their mitochondria are thrown on the Tour de France treadmill, they're not going to be able to breathe enough. They physiologically cannot respire quickly or deeply enough to blow off enough CO2 to keep their blood pH in normal physiologic range. But they can't just jump off the treadmill because they signed up for it whenever they took the drug. And so CO2 builds up, blood pH drops. If your blood pH is below 7 for any amount of time at all, you are going to die because the proteins in your body just denature. They move away from their optimum, from their PI, or from their optimum pH where they normally function, and they denature, and you can't sustain life anymore. And so don't take decoupling agents. Just work out. It's a lot safer. Also a lot more miserable. Anyway, so that's just something I really like that discussion because it allows you to think about everything across the board from electron transport chain to equilibrium. Notice how important the gen chem stuff is here, like the equilibrium idea, the solubility ideas, the acid-based stuff, right? All of that is crazy important for understanding biochemistry deeply, which is why we kind of like to start you all with that stuff first. They get good at gen chem and everything else kind of, it's like the rising tide that lifts all boats. So it's just a relatively useful discussion, and it's one that I see tested all the time. There's, I don't know, Elliot could probably speak better to this than I could, but there's a ton of stuff in the AAMC corpus that has to do with decoupling in some form or another. So just be aware that that's a possibility that can happen and know what happens. And also notice how different that is from cyanide toxicity. In one case, we're inhibiting complex 4. In one case, we're dissipating the gradient that was made. And in both cases, the problem is with the gradient. But in the case of inhibiting complex 4, we can't burn calories anymore. So we die a little bit faster. And there's not really any use for that at all. But in the case of the decoupling agent, again, acting on the same mechanism, just in a slightly different way, we can live through it and can actually get benefits from it. But we die a very different way doing that because we're dying of acidosis rather than just inability to make ATP. So just something that I think is really interesting to think through. The last thing that I have on the agenda here today is I wrote down cholesterol and statins. So again, this is my outline for this whole thing. I think we've touched everything. But I wrote down cholesterol and statins for this one. What I want to do there is think about what statins do and the effects. And now I'm kind of running it. I just realized that. What statins do and the effects that they have. So statins are drugs that most people over a BMI of 30 and over 55 years old are on. They're sort of miracle drugs because they are able to dramatically decrease your LDL levels without really having to do anything but take this drug once a day, and the side effects are minimal. I had the fortune of the two guys, Doctors Brown and Goldstein, who invented statins, were my biochem professors in med school. And so they won a Nobel Prize for this. And so it's just like we learned way too much about statins because of that. It's like a whole week of statins. So what statins do is they inhibit an enzyme called HMG-CoA reductase, which is something you do need to know. You do need to know what HMG-CoA reductase is for the MCAT, but again, it's like crazy low yield. So this is way out in the weeds. So don't worry about it unless you're already getting like 515 or higher on practice tests, maybe even 520. But HMG-CoA reductase, rate limiting enzyme for de novo cholesterol synthesis. And so by inhibiting that, your levels of cholesterol drop. Now, cholesterol is also important for a lot of stuff. I mean, many of you may know that it is the precursor for synthesis of steroid hormones in your body. And if you don't have steroid hormones, you might die also. You're going to die if you don't have steroid hormones. It's also the way that we maintain membrane fluidity in all of our cells. And so that's really important, because all of your cells need it. It's also the key constituent for bile production. So lots of important stuff that cholesterol does. So if you drop cholesterol levels to 0, that's bad. Fortunately, de novo cholesterol synthesis is not the only way that we get cholesterol. We also get it from our diet, right? And so it's a combination of those two things that lead to the total cholesterol that you have in your bloodstream. The amount of cholesterol that people have turns out to be highly genetic, and it has everything to do with the specific variant of HMG-CoA reductase that you have, or at least it seems to have everything to do with that. It might also have to do with the protein part, like the gene that codes for the protein part of the lipoprotein, right? Because all of the cholesterol is riding around on lipoproteins, so certain lipoproteins are going to have higher affinity for cholesterol than others. So a combination of those two things. So whenever you inhibit de novo cholesterol synthesis, though, and usually these designer drugs, and Brown and Goldstein did this before people had done a ton of the designer drugs, most of the designer drugs ended up in lawsuits because while they targeted specific biochem pathways that we found to be important, they also had untold effects until we got them out into a broader population. And so things like thalidomide, which was a treatment for nausea for women who were pregnant in like the 1980s, I think, thalidomide led to babies being born with lots of extra limbs or no limbs at all. And so bad thing, right? Look, these guys just got lucky, and they admitted that like in their lectures. So HMG-CoA reductase decreases cholesterol via stopping the de novo synthesis pathway, but that's just like one small thing. That also by decreasing de novo synthesis, they decrease initial steroid hormone production because you have less cholesterol. But then what happens is the body's smart enough to shunt excess cholesterol to the steroid pathway away from the other things. And so your levels of steroid hormones don't actually change at all when you take HMG-CoA reductase, but now you are using more of your excess to make steroids. And so what does happen, the one bad side effect from it is that membrane fluidity drops just a little bit, because that's not as high of a priority as steroid hormone synthesis, I guess. But when membrane fluidity drops, these people become predisposed to more rhabdomyolysis whenever they exert themselves without being hydrated. And so that's like the big side effect. Rhabdomyolysis is spontaneous degeneration or necrosis of muscle fibers. Usually only happens during intense workouts and people who don't hydrate, who take statins. So that's the negative side effect. So the third thing that cholesterol is important for, which is bile acid synthesis, this actually turns out to be just an even better feature of statins, because by decreasing de novo cholesterol synthesis, you also decrease the cholesterol that you have available to form bile acids. And so by decreasing your bile acid production, you decrease your ability to emulsify those fats in the GI tract, which causes your pancreatic lipase not to be able to access those fats as easily. And that causes more of those fats to not even get absorbed across the small intestinal lining. It does come with a negative side effect of what I've been told are greasy, really gross, unpleasant stools. However, that's better than dying of a heart attack. So, it's just like another means by which this is able to decrease your overall cholesterol. It turned out to be like a drug good enough to win a Nobel Prize. So that's a general overview of cholesterol and how to think about drug design as well, and the sorts of effects that can happen. That is everything that I had to talk about today. I hope that this was useful, at least from, again, not like memorizing the ideas, because you can do that on your own. You don't need me to just read a list of stuff. If you're going to have somebody do it, just like get on, oh shoot, what's it called? 11 Labs, and get like Liam Neeson's voice or something and upload it, and then have him read the book to you, because he will be better than me. But anyway, I'm going to go ahead and open it up for questions. I can see that there's been some stuff typed in the chat here. So let me know if there's anything that I haven't addressed, or if any of you have any questions from any of this stuff. I'm happy to answer whatever here for at least the next maybe like 15-ish minutes.